Well, thank you, Anna. And thank you also to the Hub BCD for organizing these seminars, and welcome everyone to the presentation of this tool that we call BigMap. And as you can see from the title of the presentation, this is not a specific tool for bioinformatics or computational biology, but it's more a general purpose tool that goes to the most basic exploratory data analysis. Mm. Um, Almost the grid part of what I will explain here, it's taken from this paper that we published last September after many years of effort. So yeah, you can go there for further details. And well, the tool, the BigMap, is implemented as an R package that you can find here in this GitHub repository. There is also a version in CRAN, but this version is not updated. So you better go to this repository for the most recent version of the package. This package is optimized for high performance computing platforms, but you can use it also for in your laptop or in your desktop. But it's designed for high computational cost. And its core is written in C++. We are using the package big memory because we use pointers to big matrices, and we use the packages as Snow and Parallel also because we implement parallelization, including open MPI parallelization. That means that we can use different nodes. We can use hundreds of cores to perform the computations. And the main goal of the package is basically to the visualization of high-dimensional data and the unsupervised clustering of high-dimensional data. Well, the main approach for visualization is the visualization of high-dimensional data is the embedding. And embedding is in an iterative process that starts with a random allocation of high-dimensional data points in a lower dimensional space, and then tries to update the positions to better represent the structure in the data. And here I'm showing you an example of this iterative process. You can see that the points started randomly allocated, and then iteration by iteration, the algorithm optimizes the position to visualize optimally the structure of the data. Well, an embedding algorithm is a nonlinear transformation from a high-dimensional space to a lower-dimensional space, and we use them for visualization. This low-dimensional space is typically 2 or 3. And the main driver of these algorithms is to preserve local neighborhoods. That means similarities of between data points. Similarities measured as by any measure of similarity, Euclidean distance, cosine similarity, Jaccard distance, whatever. But it is important to note that these similarities are not taken into account. That means that the embedding algorithms just try to preserve local similarities. And most popular embedding algorithms are UMAP and TSNE. And from TSNE, we have two well-known variants, the Barnes-Hut TSNE and the fast interpolation TSNE, which is the most recent and the faster. The main parameter in both algorithms is the neighborhood size, which in TSNE is called perplexity. Well, in TSNE is called perplexity. So that for small values of this parameter, the visualization, the embedding will capture what we call local structure in the data. And for higher or large neighborhood sizes, the embedding will capture global structure in the data. Both algorithms present a complex parameterization with many parameters that probably are familiar for all of you that have been using these algorithms, the learning rate, momentum, exaggeration, many others. And then they are complex, they are computationally expensive algorithms because they have complexity of order quadratic in the data set size. But both UMAP and TSNE achieve a low computational cost when they use small neighborhood sizes. Well, how is the embedding implemented in BigMap? BigMap implements the PTSNE, which is a parallel implementation of the Barnes Hub TSNE. With the benefits that the parametric configuration is really simple because we have developed self-adaptive schemes for learning rate and momentum, and we drop early generation. We do not use it. So this simplifies the fine-tuning of the parameters of the algorithm. Also, while UMAP and TSNE and PTSNE claim robustness to the neighborhood size parameter, which is something that is somewhat counterintuitive, because what we would expect from an embedding or visualization algorithm is to be like kind of lens that can show us different resolutions or different scales of the structure. PTSNE is really sensitive to perplexity, so it can show us these different levels of structure in the data. Also, it scales well with perplexity, and these two features together result in that PTSNE allows exploring a structure across scales. Well, one downside in the current version of BigMap is that by now we can only perform embedding into dimension. So at this point, it is really important to understand what do we mean when we talk about local and global structure in the data. And for this, I will show you an example based on this structure, which is the Sierpinski 3D triangle. This is a fractal structure. And so if we were to perform an embedding of this structure, we would expect that when we use a low neighborhood size parameter, we would expect to capture the little tetrahedrons that form the structure, to visualize them. And as we move to higher values of perplexity, we would expect the algorithm to capture the global structure of or higher levels of structure present in this data. And this is what we see here. Here we have used PTSNE with a very low perplexity value, just 1% of the total of nodes in this graph. And we can see that what the PTSNE visualizes is these little structures that would represent is the visualization of these little tetrahedrons. But as the neighborhood size is low, it does not convey enough information for the algorithm to reconstruct the structure at a higher level of organization. But then if we move to higher levels of perplexity, here, for instance, we have used 5% of the data, then we can see how these little structures start to organize in higher level structure, higher level structures. And the same as we move to higher levels of higher values of perplexity, well, we see that as we increase them, we recover different or the structure present in the Sierpinski 3D at different at different resolutions. And here, for instance, we have moved up to 90% of all the data, and what we capture, yeah, really, well, all the little structures that we were seeing here get connected, all them together, showing the whole structure of the pyramid. And finally, we have this, in which we have used 95%, and we can clearly see the four faces of the tetrahedron. Well, That means that the embedding algorithms need this ability of this scaling, computational scaling, not only for the dataset size, but also related to the neighborhood size. And here I'm showing you another example. This is a dataset representing the prime factorization of the first million integers. And this is a sparse matrix with 1 million rows, well, one less because the integer 1 is not there, and about 80,000 dimensions, which are the number of prime factors that lay within this range of the first million integers. And here we have a visualization with a low perplexity, and here we have a visualization with a high perplexity, really high. In this case, we are using 100,000, a neighborhood size of 100,000. And we can see here that for, well, the different powers of the prime factors are really organized, something that is not present here. But here we can see it. Here we can see the powers of 2, the powers of 5, powers of 3. And here between 2 and 5, we see the 10. Between 2 and 3, we get 6. Here we have the 7 and the powers of 7. And here we have prime numbers, which have more, not so much integers that are powers of them. And finally, we here have the highest prime number. So in this area, we would have all these prime factors that do not, that there are no integers that are factorized with them within this range. No? And for this visualization, I'm comparing the running times with FIT-SNI here and with PT-SNI here. And you can see that with FIT-SNI, we computed different visualizations with 50, 100, 200, 300, 400. And we can see how the running time rapidly scales exponentially. While with PT-SNI, we were using 1,000, 5,000, up to 100,000 for perplexity. And the running time scales more linearly. How do we achieve this with PT-SNI? Well, PT-SNI implements a real parallelization of the TSNI based on what we call the chunk and mix protocol, which simply means that we take the data set and we chunk it, and then we start independent instances of the TSNI in independent threads with each chunk. Then there is a master process that pulls the partial mappings that come out from these independent threads, mixes them, rechunks them again, and starts the process again. This chunk and mix process is done for several epochs and until it converges. And the key to convergence is that when we chunk the data set, we do not send one chunk to each thread, but we make the threads share some chunks. And this generates a link between all the threads that is what allows the whole process to converge to a global solution. The BigMap also presents some or offers some tools for the assessment of the quality of the embedding. First, we have what we call the embedding cost size function. Well, TSNI minimizes a cost function, which is defined as the Kullback-Leibler divergence between distributions P and Q, that are distributions that explain the distribution of neighborhoods in the high dimensional space and the low dimensional space. And TSNI tries to match these two distributions by minimizing this function. When the PT-SNI starts, it performs a random, well, it starts with a random embedding on a unit disk with diameter 1. So we start here, and we use pseudo cost, which is a normalization version of this Kullback-Leibler divergence, which is always around 1 when we start, no? And then as the optimization proceeds, the embedding area expands here to better accommodate the data, and the cost function is minimized and flattens as the optimization reaches a local or a global minimum. So as long as we observe a plateau in this in the last epochs, then we know that the algorithm has converged to a global solution and to a minimum. Another tool for a quantitative assessment of the more quantitative assessment of the quality of the embedding is what we call the CARI neighborhood preservation. This is a measure of matching between the high dimensional and the low dimensional neighborhoods for the whole range of neighborhood sizes. And for instance, here you can see these blue lines that show these correspond to a low perplexity, and they show high matching for small neighborhood sizes. But for large neighborhood sizes, the matching is really poor. But as we increase the perplexity, we gain representation of these these larger neighborhoods, and we lose representation of the small neighborhoods. So the area under these curves summarizes the level of matching, and BigMap shows us two versions of this plot, a linear and a logarithmic one on the neighborhood sizes, so that we can better assess what is happening in this area with the local neighborhoods. No? We can see that there is a trade-off between large neighborhoods and small neighborhoods. And depending on the value of perplexity, we capture better ones or the others. To put BigMap in the context of the hub, I will show you a few examples with transcriptomic data sets. You can find these in the paper, so for further details, you can go there, but I will quickly explain just the idea, which is that in this case, we have a data set with cells from adult mouse cortex. Here we have three groups of neurons, inhibitory neurons, shown in warm colors, excitatory neurons, shown in cold colors, and non-neural cells, shown in brown-gray colors. And if we perform a visualization with the two first principal components, we see that these three groups can be clearly separated. Then Kovac et al. In this paper, The Art of Using TSNE for Single-Cell Transcriptomics, probably most of you have read it and you know it. It's published in Nature Communications. They show how to fine-tune the TSNE so that they achieve this visualization of the structure in which we observe the global structure, because we have here all excitatory neurons. In this part, we have here all the warm inhibitory neurons, and here we have the non-neural cells. And in each group, we get these subgroups that correspond to the different subtypes of cells, neurons. And with PTSD and a low perplexity, we recover these subgroups, although we do not observe the global structure here. But if we go to higher perplexity, then we recover the structure at a large scale, the global structure, to the point that we basically get what we get with PCA, but we still observe these subgroups. We can observe these subgroups of types of cells. This is another example with a data set with cells from the mouse retina. And well, it is exactly the same and the same idea. Okay, let's talk a little bit about clustering. Unsupervised clustering is performed with BigMap in these two embeddings. So we do not perform clustering on the high dimensional space. The idea is to first perform the embedding, and then once we have the representation in 2D, Then we perform the clustering there. The clustering consists in two steps. In the first step, we build a kernel density over the two-dimensional embedding. And for this, we use a perplexity adaptive kernel density estimation. This is an algorithm that we specifically developed in BigMap. And we are using the same approach that we use to perform the embedding from the high dimensional to the low dimensional. So we use perplexities to determine adaptive bandwidths for the kernels that we use on each data point to build the kernel density estimation function, which is shown here with contour colors. No? And then once we have built the kernel density, then we run a water track transform over this density landscape, which is a function that follows the water tracks in this density landscape and allows us to partition this embedding into different parts and to assign clustering labels to each data point. And then we can use these clustering labels to visualize the clusters, to overlay the clusters on the 2D embedding. Well, this data set that I was showing here is a real data set that forms part of a working preparation here in CEAB, some researchers here in CEAB. And it's basically a data set with counts of presence and absence of different genes in different genomes. No? And we are trying to find genomes that share the same genes. So how do we go with to cluster this? First step will be to compute the embedding at different scales. You see here that we have computed it with low perplexity from 0.5 to 10% of the dataset size. We have the cost and size functions. They all look smooth, so we reach a good solution in each case. And these are the embeddings that correspond to each one of these perplexities. Then how do we choose the perplexity value that we should use, or the neighboring size that we should use to perform the clustering? Well, for this, we use the Carry neighborhood preservation here, because these curves show maximums that correspond to neighboring sizes for which the preservation of the neighborhoods is maximum between the high and the low dimensional space. So it makes sense to use these neighboring sizes. No? So for this embedding, which is with the lowest value, the one that is showing the most local structure, and that is best or most probably reflecting the clustering at the highest resolution level, we have a maximum of KMP at 305. So we use this value of perplexity. Be careful not to confuse these two values of perplexity. Here we use the perplexity to make the embedding from the high dimensional space to the low dimensional space, and afterwards we choose a different perplexity to determine the bandwidths here to construct the kernel density estimation. But the approach is the same. Perplexities or neighborhood sizes considered in a high dimensional space and in the low dimensional space. What makes the whole BigMap procedure coherent. So we make the clustering using this value of perplexity, and we get these labels, these clusters. Then what we do is that we can map these clusters, we can overlay these clusters on the embeddings that we have done with higher perplexities. And we observe that, for instance, these two groups that have been clustered together, we can see how when we look at the structure at higher scales, we can see how they move apart. And they end up here when we have a good representation of the global structure. And so that means that they should correspond to examples that are not so similar as we may think when we find them here together. And then we can do the inverse. The inverse thing. So we can compute the clustering on the embedding that is showing the global structure at the highest perplexity. So we take this embedding now. Look, we first made the clustering here. Now we are doing it here. And with this clustering, we get different labels. And then we can map these labels, or we can trace this clustering backwards to the lower perplexity embeddings. And then we observe things like, for instance, this blue cluster here that ends up being as two different clusters here that might seem here as something different, but they are not so different when we observe them here, looking at the structure at the global structure, and the same with this other cluster. We have these two or these several groups here that may look here as something different, but when we look at them here, we should conclude that they are not so different as they may seem. And finally, what we can do also with BigMap is to overlay metadata on the embedding so that we can check how things have been mapped. And here, for instance, we are mapping different taxons, different groups of genomes, and on both, on the local mapping and on the global mapping, and we can see whether they have been mapped together or far apart one from the other. So we can check these kind of things. And well, that's basically well, a very fast explanation of what BigMap can offer. I hope it was clear enough. Just to, I hope I was good in giving you a first picture of what the tool can do. And if you have any questions, I will be happy to answer you.